In this lecture, we will discuss about Southern blotting. Southern blot is the analytical technique that uses in molecular biology, immunogenetics, and other molecular methods to detect or identify specific DNA of interest in a mixture of DNA sample. This technique was first developed by a molecular biologist E.M. Southern in 1975 for analyzing the related genes in a DNA restriction fragment and thus named as the Southern blotting in his honor. So in Southern blotting, first the DNA containing the desired gene of interest is first extracted from human cells and then it cut into fragments by restriction enzymes, and next the fragments are separated according to the size by gel electrophoresis, and here each band consists of many copies of a particular DNA fragment, and the bands are invisible but they can be made visible by staining. And in next step, the DNA band from the gel are transferred to a nitrocellulose membrane by blotting, and the solution passes through the gel and filter to the paper towel, and then they produce a nitrocellulose filter with DNA fragment positioned exactly as on the gel. And in next step, we expose the filter to a radioactively labeled probe for a specific gene, and this probe will base pair or hybridize with a short sequence present on the gene. And then the filter is exposed to X-ray field. The first step is DNA isolation. In DNA isolation, suppose we have isolated genomic DNA from bacterial cells, and we know that this DNA contains thousands of genes or DNA sequences encoding various proteins in bacteria. But we want to find out the presence of a specific DNA sequences in the bacterial genome, or in other words, we are searching for a gene or DNA sequence of interest whose nucleotide composition is already known to us, but we want to find out whether this DNA sequence is present in the bacterial genome or not. So after DNA purification, we will perform restriction enzyme digestion. In restriction enzyme digestion, the restriction and the nucleases enzymes are used to cut high molecular weight DNA strand into smaller fragments. One or more restriction enzymes can be used to achieve such fragment. After restriction enzyme digestion, the separation can be done by gel electrophoresis or agarose gel electrophoresis, in which negatively charged DNA fragment moves toward the positively charged anode, and the distance moved depending upon its size. So after gel electrophoresis, the DNA then denatured with a mild alkali such as sodium hydroxide solution, and it causes the double-stranded DNA to become single-stranded and making them suitable for hybridization. After denaturation, the denatured fragments are then transferred onto a nylon or nitrocellulose filter membrane, which is done by placing the gel on top of a buffer saturated filter paper and then laying nitrocellulose filter membrane on the top of the gel. So finally, some dry filter paper are placed on the top of the membrane and fragments are pulled towards the nitrocellulose filter membrane by capillary action, and the result in the contact print of the gel. Then after blotting, the nitrocellulose membrane is removed from the blotting stack, and the membrane with single-stranded DNA attached to it is baked in a vacuum over 80 degrees C for 2 to 3 hours or exposed to ultraviolet radiation to permanently attach the transferred DNA onto the membrane. So after blotting, the hybridization is performed In hybridization, the membrane is exposed to the probe, which is single-stranded fragment with the specific sequence whose presence in the target DNA is to be determined. The probe DNA is then labeled so that it can be detected, usually by incorporating a radioactivity or tagging the molecule with a fluorescent. Then after hybridization, the membrane is thoroughly washed with a buffer to remove the probe that is bound non-specifically or any unbound probes present. Then the hybridized region are detected autoradiographically by placing the nitrocellulose membrane in contact with the photographic film, which shows the hybridized DNA molecule. And the pattern of hybridization is visualized on X-ray film by autoradiography. And here is the final autoradiogram that obtained after southern blotting is something like that. Now let's summarize southern blotting steps. The first is DNA purification. After DNA purification, then we perform restriction enzyme digestion, and then the fragments are separated through gel electrophoresis, and then they are denatured and we do blotting, and then we perform hybridization, and finally we wash to remove the unbound probes, and then we perform autoradiography. Well, southern blotting is used in different applications, like they are used to identify specific DNA in a DNA sample and preparation of restriction fragment length polymorphism maps, and they are also used to detect mutations and deletions or gene rearrangement in DNA. And they also help to find criminal cases and DNA fingerprinting. And they are used in detection and identification of transgene in a transgenic individual. And with the help of southern blotting, we can map the restriction site, and it also used for diagnosis of infectious diseases and prognosis of cancer, and parental diagnosis of genetic diseases can be confirmed through southern blotting. And it also helps to determine the molecular weight of a restriction fragment and to measure relative amount in different samples. So it's all about southern blotting.